Okay, we'll start the live stream now. All right. Well, good afternoon. Thank you for coming and welcome to the biology keynote session of our second annual Oxford-Cambridge Student Symposium, Varsity Sci. I'm Shreyas, president of the Cambridge University Biological Society, and today it's a great honor and privilege to host Professor Randy Schekman as our keynote speaker today. Before I introduce Professor Schekman, I have a few words from our sponsor, BitBio. Today, we have a unique opportunity to build a bright future. To do so, we need to extend our technical repertoire and transition biology to engineering. Tapping into the potential of biology will accelerate biomedical research, enable new generations of cures, and open up new avenues for increased global sustainability. Founded by Dr. Mark Cotter and Florence Schuster, BitBio is an award-winning human synthetic biology enterprise. BitBio's mission is to code cells for health. To do so, they apply the principles of computation to biology. Their current focus is to develop a scalable technology platform capable of producing consistent batches of every human cell. Now, coming to our fantastic speaker, Professor Schekman is an investigator of the Howard Hughes Medical Institute and a professor of cell and developmental biology in the Department of Molecular and Cell Biology at the University of California at Berkeley. His work has attracted many accolades, including election as a member of the National Academy of Sciences in 1992, the Albert Lasker Award for Basic Medical Research in 2002, and most notably, the 2013 Nobel Prize in Physiology or Medicine for discoveries of machinery regulating vesicle traffic, a major transport system in our cells. Professor Schekman is also devoted to the promotion of science in as open a manner as possible. He is former editor-in-chief of the Proceedings of the National Academy of Sciences, and in 2011, he was appointed as editor of eLife, an open access journal published by HHMI, Max Planck Society, and the Wellcome Trust. Today, he'll be speaking to us about selective protein sorting into exosomes, a role in cell differentiation, and a possible tool in genome editing. This talk will be followed by a Q&A session. If you're on YouTube, please post your questions in the chat throughout the talk. If you're on Zoom, you can either ask your questions in the chat, you can send them privately to me, or you can use the raise hand function, and I will let you unmute and ask the question to Professor Schekman himself. Please note that you will be on the recording. So without much further ado, I'd like to welcome Professor Randy Schekman.
Thank you very much, Shreyas. It's a delight to join you this evening. It's now early morning in Berkeley, where it's supposed to be up to 30 degrees centigrade, so it'll be a lovely day here. Of course, I'll be indoors all day, so I won't enjoy it as much. It's always a pleasure to visit with students at Cambridge. I've had other opportunities to do so over many years, various seminars. My first trip to Britain was to spend my third year as a university student in Edinburgh, where I learned a great deal about British culture. It was
sorry to interrupt, Professor Schekman. Your screen isn't being shared. If you'd like to just reshare your screen again, sorry.
You can't see me, or you can't see my slides?
I can't see your slides.
I haven't started my slides
yet.
yet.
I'm so sorry. I just assumed you already had. Sorry.
Okay, anyway, let's get to business. So today I'm going to talk about fairly recent work in my lab, having to do with a process of vesicle traffic that is devoted to the export of proteins and small RNA molecules in extracellular vesicles or exosomes. So let's begin with just a brief summary of what we know about extracellular vesicles. First of all, they're produced by almost all eukaryotic cells, frankly, even prokaryotic cells. But most of what we know has been learned with cells grown in culture, mammalian cells grown in culture. There are two paths that are known. One is to produce small, what are referred to as microvesicles that bud from the plasma membrane. They capture proteins and RNA, similar to that employed by enveloped RNA viruses for their production. Another more elaborate path relies on an endosome that can capture also proteins and small RNAs, in this case fairly selectively, by budding into the interior of the endosome, which over time accumulates a population of intraluminal vesicles. Much of the material so captured is delivered to the lysosome, where these small vesicles are degraded. But some fraction of the structures fuse at the cell surface and discharge a bolus of vesicles. There's a great deal of interest in what these vesicles may do. They could either be another means of disposal to get rid of things that the cell doesn't want, but there's some evidence, emerging evidence, that these vesicles can be internalized by target cells, probably fairly selectively, and where they can deliver their content by fusion to discharge the luminal material, RNA and proteins, into the cytoplasm of the target cell. I'm going to give you two examples of that, where we have evidence in one situation that these vesicles can be functionally taken up and can influence the rate of differentiation of embryonic stem cells into neural progenitor cells. But then I'm going to turn around and tell you a very different story, where we've attempted to harness these exosomes to capture genome editing functions, specifically Cas9 in a guide RNA. And you'll see that, at least in these circumstances, exosomes seem not to be have any functional uptake, but we've turned the story into something rather different and interesting, as you'll see, so bear with me. Now, over the last few years, we've devised means of purifying extracellular vesicles by separating them on a buoyant density gradient. This is one example that we published a couple of years ago through the efforts of a very talented graduate student, Moraima Timosh-Diaz. She took extracellular vesicles from a breast cancer cell line called MB231. You'll hear more about that cell line later on. And just the crude collection of vesicles can be resolved by sedimentation on a buoyant density gradient, where she produced a population of low buoyant density vesicles that are marked by these proteins that are found in extracellular vesicles, but some apparently are very selective for the low buoyant density fraction. In contrast, there's a high buoyant density fraction, including a traditional marker of exosomes called CD63. It's a 4-spanning membrane protein, and you can see that this protein is highly selectively enriched in these high buoyant density vesicles. In contrast, the culture medium in which these vesicles are secreted also contains proteins, likely large ribonuclear protein particles, that include editing functions for microRNAs, AGO2 and dicer, but these are not in vesicles. They sediment all the way to the bottom of the tube, so they're not enclosed within the vesicles. Now, much of the work that we've done over recent years has been to explore the content of these high buoyant density vesicles. I won't tell you about that today, other than to summarize by saying that certain microRNAs are very efficiently sorted into these high buoyant density vesicles. And we've been exploring, using a biochemical approach, how that sorting may occur. But I'm going to turn my attention to more recent work that was, some of which was only recently published, the efforts of a postdoctoral fellow, Lu Song. Lu started the lab with experience in the differentiation of nerve cells from mouse embryonic stem cells. And we asked whether 3 different conditions that allow the differentiation of nerve cells in cell culture allow us to explore the content of extracellular vesicles, some of which may be secreted by the cell body, and others of which may be delivered by fusion of a multivesicular body at the nerve terminal. We made several observations that convinced us that something different was going on during the course of differentiation of these cells. So let me tell you about the 3 procedures that we've used to differentiate mouse embryonic stem cells. One way is to treat cells with growth factors, certain cell lines with growth factors. One such condition is treatment of a neuroblastoma mouse cell line that differentiates into neural progenitor cells when the cells are treated with retinoic acid. So you see, after several days, these nerve-like projections emanating from these cells. Another that you don't see here is when another mouse cell line called PC12 is treated with nerve growth factor. That also produces a response similar to that seen with retinoic acid. And a perhaps more authentic means of differentiation has come when we use mouse embryonic stem cells themselves, and in a two-step procedure, differentiate them first into embryoid bodies. Cells from that are dispersed and grown in a selective medium for the next 4 days, during which time they produce, again, neurite projections and look very much like, and as you'll see, express genes that are characteristic of neural progenitor cells. Now, one of the ways that we've evaluated this, of course, is by isolating the vesicles using a buoyant density gradient. We used a different one that I mentioned earlier because we found that during the course of exposure of these mouse cell lines to hormones or conditions of differentiation in several stages, we found that the vesicles that are produced change their behavior in buoyant density. So we've collected a rather broad group using a procedure shown here where the vesicles are captured by high-speed sedimentation, are suspended in a high concentration of sucrose, and then floated up through a layer of 40% sucrose where the vesicles equilibrate at the boundary between 20 and 40%. These vesicles are then diluted, and all the experiments I'll describe have been done with this material. Well, one of the first early surprises was that in the course of this differentiation regimen, these cells produce an increased number of extracellular vesicles. Here's an experiment with nerve growth factor treated PC12 cells, and during the course of 9 days, we observed a threefold increase in the production of extracellular vesicles. Here shown quantified in a particle tracking device that measures the average diameter, and this population average is somewhat below 200 nanometers in diameter. More strikingly, when M2A cells are treated with retinoic acid, we observe a substantial, more robust 14-fold increase in the production of these vesicles, quantified here using this particle tracking device, but also evaluated by the content of these vesicles of proteins that are characteristic of extracellular vesicles. So you see during the course of this 6-day differentiation, a huge increase in this protein, another force-spanning membrane protein, but also in the protein that I mentioned before that is characteristic of exosomes called CD63. So something is going on in these cells that has promoted the production of bona fide exosomes and a large pool of extracellular vesicles. So the critical question has been, what are these vesicles doing? And so what Lou did was to take naive mouse embryonic stem cells and to initiate a program of differentiation by simply transferring them to serum-free medium. And during several days of that differentiation, she can observe the expression of genes that are characteristic of the neural progenitor cell. Here are two, nestin and 6-3. But more interestingly, and that's shown with just no vesicles in the light green, there's an increase in the expression of these genes. What was more interesting was in the presence of a fixed level of extracellular vesicles produced from PC12 cells treated with nerve growth factor, she observed about a 2-fold increase in the rate of expression of these genes over the background. Now that modest 2-fold increase is enhanced if the ratio of extracellular vesicles to neural progenitor cells, differentiating mouse embryonic stem cells, is increased. And so she was able to observe perhaps a 3-fold increase in the expression of these two genes dependent upon vesicles, specifically upon vesicles that had been exposed to nerve growth factor, PC12 cells. So something is in these vesicles allowing them to interact with mouse embryonic stem cells to speed the pace of differentiation. So the experiments I'll tell you about now address what that may be in these vesicles. And we've identified one, possibly two key proteins in these vesicles that may be responsible for this enhanced rate of gene expression. We observed in the course of evaluating the protein content of exosomes, or EVs, versus cells in the course of this 9 days of differentiation, that a couple of proteins in particular, regulators of cell cycle progression, called cyclin D1 and CDK4, which form a complex that guides the transition from the G1 to the S phase of the cell division cycle. We found that these two proteins were fairly selectively packaged into EVs from PC12 cells treated with nerve growth factor, whereas other cell cycle proteins that control other transitions in the cycle seem not to be packaged at any measurable level. This flotilin is a marker protein characteristic of EVs, just to show that this protein is clearly contained in the vesicle fraction. So this was quite interesting. Perhaps these are proteins, cyclin D1 and CDK4, that influence the pace of neural differentiation when they are delivered from an exogenous vesicle. Well, the same thing was found for mouse embryonic stem cells that were themselves carried through the path of differentiation that I told you about, where after 8 days they form embryoid bodies. They are then dispersed and allowed to differentiate themselves into neural progenitor cells. And so Lou sees quite a robust capture of cyclin D1 into these EVs after this 12-day period. And perhaps most importantly, these vesicles from the 12-day differentiation, but much less so from the 8-day embryoid body differentiation, these vesicles produce about a 3-fold increase in the expression, again, of 3 markers of neural progenitor progression. So now the question is, where are these proteins in respect to the vesicle? Can we be a little bit more specific? Are they carried on the surface of the vesicle, or are they sequestered in the interior of the vesicle? And for this, a standard experiment that one uses is to expose the vesicles, the purified vesicles, to exogenous proteinase, in this case proteinase K, in the absence or the presence of a detergent, Triton X100, a mild non-ionic detergent, to see whether the protein is inaccessible to exogenous protease unless the membrane is dissolved by detergent. And you can see quite clearly that all of the proteins, including cyclin D1 and CDK4, plus the standard markers of the vesicle, are resistant to a fairly high concentration of protease unless you add detergent, which permeabilizes the membrane and renders the internal contents accessible. Now, further, Lou could show by selective immunoprecipitation using an antibody against an exposed epitope of the CD9 protein, that those vesicles carrying CD9 also carry cyclin D1, whereas a control antibody, not recognizing any exosomal or vesicle protein, when fixed to beads, does not precipitate this material. So we conclude from this experiment that cyclin D1, CDK4 are inside of vesicles that carry bona fide extracellular vesicle marker proteins. Now, this experiment was an attempt to test the specific role of cyclin D1 in accelerating the pace of differentiation. So we did 2 experiments. One was to use N2A cells in which the cyclin D1 gene was introduced by transient transfection so as to overproduce the protein about 3-fold in the cells, resulting in about a 2-fold increase in cyclin D1 content in the isolated vesicles. So then we asked, with these vesicles, does the presence of 2-fold more cyclin D1 further enhance the rate of expression of the neural progenitor genes? And you see in this data that both that PAX6, 6-3, and MAP2 are enhanced by the overexpressed cyclin D1 in relation to that seen with just the endogenous level of cyclin D1 produced by retinoic acid differentiation of the N2A cells. The converse experiment was also possible. It's possible to knock out the cyclin D1 gene from N2A cells. And you see with isolated cells or isolated vesicles, only a shadow of some signal of cyclin D1 by immunoblot. Perhaps one of the paralogs of cyclin D1 is somewhat cross-reactive, but largely cyclin D1 is gone from these exosomes. And sure enough, when you look at these exosomes, they are much less active than the exosomes from retinoic acid treated normal cells. A reduction again in all three of these marker proteins, indicating that at least cyclin D1 is a rate limiting component in this differentiation process. There likely are other proteins that are required that may also be rate limiting, but this is at least one of them. Now, the final question for this part of the story is to ask, is there a way that we can demonstrate that cyclin D1 contained inside of these vesicles is taken up and delivered functionally into the mouse embryonic stem cell so as to enhance the rate of differentiation? For that to occur, the vesicle has to fuse. It either could fuse at the cell surface or by analogy to enveloped RNA viruses, it could be internalized where the vesicle would fuse with the endosomal membrane, and on fusion, the luminal content of the vesicle would be discharged into the cytoplasm of the cell, and cyclin D1 may then migrate into the nucleus where it could enhance gene expression, likely indirectly by targeting other proteins. So let me show you the experiment that Lou did that gave us some confidence that these vesicles were taken up and that the cyclin D1 was exposed functionally into the target cell. The way she did this was to use a now quite popular technique called proximity labeling, where an enzyme called APEX, the gene of APEX, is fused to the cyclin D1 gene to make a bifunctional fusion protein that can be packaged, and we showed in other experiments, this can be packaged into extracellular vesicles. And if the vesicles are incubated by themselves, as you'll see in a control experiment in a moment, they label proteins that are internal to the exosome. But if you expose these vesicles to cells and allow the vesicles to be taken up, and then add the reagents, biotin phenol and hydrogen peroxide, that are required for the APEX enzyme to biotinylate neighboring proteins, we could then ask by difference, are there some new proteins that have been labeled in the acceptor cells dependent upon the cyclin D1 APEX that had been delivered? So here's the first experiment that characterizes the biotinylation of endogenous proteins in these cells. So the experiment is conducted in two controls. One is where the cells, having been allowed to internalize these vesicles, are incubated after several days, briefly with just hydrogen peroxide, or with just biotin phenol, neither of which is sufficient for the APEX enzyme to catalytically biotinylate neighboring proteins. But when both are present, Lou observed a subset of lower molecular weight proteins that can be detected when the gel is blotted with streptavidin, which binds to biotin, tagged with an enzyme, horseradish peroxidase, that gives a visual signal. So this lane shows a set of proteins that have become biotinylated, dependent on the exogenous vesicles and these reagents. Now, it's obvious that there are other proteins of lower mobility, higher molecular weight that are abundantly labeled even in the absence of these reagents. And we guess that these proteins are endogenously biotinylated proteins that are characteristic of other processes in the cell. So we focused our attention on those proteins that are unique, that become biotinylated uniquely in this one experiment with both substrates present. These proteins were purified by affinity binding to streptavidin beads and subjected to proteomic analysis. Among those proteins that become selectively biotinylated, Lou found a number of RNA binding proteins. Now, this is often true of, it's often characteristic of exosomes that they capture RNA and RNA binding proteins. she found two that I'll tell you about in a moment that were only found when the vesicles were internalized by target mouse embryonic stem cells, but were not found in the vesicles themselves. These two proteins are called Lin-28 and nucleolin. Both of these are nuclear proteins, and both have been implicated in cyclin D1 control of cell cycle progression. Now, first of all, and importantly, if the vesicles carrying this fusion protein were subjected to the regimen of hydrogen peroxide and biotenphenol, and the proteins that were biotinylated were isolated by streptavidin beads, and then run out on a gel, neither Lin-28 nor nucleolin are detected by immunoblots. So these vesicles did not carry these two proteins. On the other hand, after exposure of cells to these vesicles for several days, she observed that only in the instance where the vesicle, the mixture of cells and vesicles was incubated with the two substrates for biotinylation, was she able to then isolate on streptavidin beads these two proteins that are then detected by a blot specific to these two proteins. So that told us that the cyclin D1 apex fusion protein must have gained access to the target cell and managed to biotinylate these two proteins endogenous to the target cell. So I'll leave you then with a simple model. There's obviously much more to do to understand what's going on here, but we believe that these model cells, or even normal mouse embryonic stem cells, when allowed to differentiate, have that differentiation speeded by the production of extracellular vesicles that are enriched in a couple of cell cycle regulatory proteins. And when these vesicles are then exposed to native mouse embryonic stem cells, the speed at which that differentiation occurs is enhanced several-fold. Now, what this may mean is that in the course of differentiation in the niche where these neural progenitor cells are being produced, the presence of other differentiating neurons may be a form of communication to enhance the process locally. And this may be achieved by these vesicles. So this is one niche, we believe, in which extracellular vesicles may have a functional role. Now, extrapolating from that, we hoped that this might be a tool that could be used to enrich other kinds of proteins in extracellular vesicles for other applications, such as in delivering genome editing functions, specifically to target cells in a whole organism. But let me, before I tell you that part of the story, first show you the group. I'm going to show you this picture later at the end, but this is my research group a few years ago at a retreat. I'm going to talk specifically now, I'm going to mention specifically the work of Moraima Timosh Diaz, a Peruvian graduate student in the lab, who did much of the work that's already been published, characterizing the highly selective capture of microRNAs into high buoyant density vesicles from this breast cancer cell line, MB231. The work that I've just described, which was published a couple of months ago in the Journal of Cell Biology, was done by Lu Song, who had a great deal of experience in mouse embryonic stem cell differentiation, and so she applied it quite nicely in the work that I've just described. I'm going to turn now to the effort that I just introduced, which has been done by this postdoc, Songyan Zhang, who has, as you'll see, successfully designed a procedure to enrich Cas9 into extracellular vesicles. And he's used that to explore the functional uptake of that genome editing protein. All right, so here's the strategy that Songyan used to enrich Cas9 and guide RNA into vesicles that are internalized into multivesicular bodies. So one can express Cas9 in a donor cell Cas9 in this case has been fused to a protein called streptavidin binding protein. It also, as you'll see, has been fused to GFP so that it can be visually examined. In the same cell, we've created a fusion to the CD63 protein. Remember, this is a four-spanning membrane protein. It's on the endosomal surface and is internalized from the endosome into intraluminal vesicles. It's possible to fuse a protein to the C terminus of CD63. The C terminus faces the cytoplasm, and so Song fused the gene for streptavidin. Now, the point of doing this is that streptavidin binding protein interacts with streptavidin, and that interaction is reversed on addition of biotin. So one can tether this fusion protein to CD63 in a reversible manner. And the hope was, and what we achieved was, that that Cas9 so tagged can be enriched along with CD63 into these vesicles. And we know that CD63 is itself very highly and selectively sorted into intraluminal vesicles. So then the guess is that once these intraluminal vesicles fuse at the cell surface, one would secrete into the growth medium a population of vesicles that has an enriched supply of not only of CD63, but of the Cas9 and guide RNA. And let me show you the data that demonstrates that that is so. So what Song did was to construct such a donor cell, grow it for several days in natural growth medium with serum. The vesicles are collected by high-speed sedimentation and then subjected to flotation, as I described earlier, by a procedure similar to what Lu Song did. All of the vesicles that equilibrate at the 40 to 10% interface are harvested and concentrated and then evaluated for the content of Cas9. So in two controls where donor cells were constructed with Cas9 tagged either with His or with streptavidin binding protein, but without a streptavidin CD63 fusion. In neither case was it possible to detect the Cas9 protein with an antibody against one or another tag. So that says that during the normal course of event, when Cas9 is being expressed in these donor cells, it is not at any measurable level passively packaged into these vesicles. In contrast, when the two fusion proteins, streptavidin binding protein and streptavidin, are co-expressed, then Song saw, at least by immunoblot, a clear enrichment of the Cas9 protein in these vesicles. So he has achieved sorting. In other experiments, he could show that when the vesicles are exposed to biotin, the Cas9 streptavidin binding protein is released into the luminal interior of the vesicle. Now, looking at the RNA, we constructed a guide RNA whose target, I'll show you in a moment, expressed along with Cas9 in the donor cells. And again, if the Cas9 is not tethered to the membrane, this guide RNA is not demonstrably captured into vesicles. Whereas when the two fusion proteins are expressed together, there is a 15-fold increase in the level of the guide RNA detected by PCR. So this worked. That was very encouraging. We have a tool now. We know from other experiments that the Cas9 was actually enzymatically active in the inside of the vesicle. So the stage is set now to try to measure its effect on a target cell. So we set up a reporter with a very sensitive readout that would allow us to detect Cas9 internalization. The readout consists of two different luciferase genes, firefly luciferase and NLuc. This cassette has NLuc stopped by the presence of a stop codon and a single nucleotide frame shift mutation, either of which is enough to block the expression of NLuc in these cells into which this cassette has been integrated. Now, the idea is that if Cas9 and the guide RNA have access to the Cas9 target by editing and repair. Some fraction of this N-loop gene will be restored to a proper reading frame, allowing the enzyme to be detected. Now, these two enzymes have different substrate specificities, and so they can be assayed independently in the same cell. So in this experiment shown on the right, if two different guide RNAs that are capable of this editing and Cas9 are introduced into this reporter directly, we observe about a 100-fold increase in the ratio of expression of N-loop to F-loop. But it absolutely depends on the expression of the guide RNA in this reporter cell. So this says we have a sensitive reporter with a very nice signal-to-noise ratio that we can use to detect functional Cas9 delivery. So now to the key experiment and the surprise. When the Cas9 guide RNA cassette is expressed directly in the acceptor cell, we saw in this case about a 70-fold increase in the ratio of N-loop to F-loop. In a control, the ratio of these two enzymes is set to 1. However, when cells are incubated with Cas9 enriched and guide RNA enriched vesicles in a ratio of as much as 100,000 vesicles per cell, Song and observed only a very moderate 50% increase in the ratio of N-loop to F-loop. Really rather surprising, and I will confess disappointing. Nonetheless, there is a measurable dose response, so we saw an increase in the level of this expression. But again, it's very modest. So we were forced to consider one of two possibilities. Perhaps the vesicles, when we isolate them on a buoyant density gradient, are fragile and they lose their ability to be taken up. Well, there's a simple test for that. What we did was we took conditioned medium from cells that were secreting these extracellular vesicles for either 3 days or 8 days. And we took that medium and simply grew the target, the receptor cell, in the presence of that medium with no fractionation of the vesicles at all. And once again, a very moderate signal, maybe up to 2-fold, still very moderate compared to when Cas9 is expressed directly in the donor, in the target cell. Now, the next experiment that gave us serious pause was even more gentle, and that was the idea that perhaps these vesicles are simply ephemeral and they don't last very long at all, whether you purify them or not. So we set up an experiment where the donor and acceptor cells are co-cultured in a chamber called a transwell chamber, where the medium contains one cell type layered on top of a membrane permeable filter that would allow vesicle traffic back and forth, and under which another cell type is grown, in this case, the donor and acceptor cell type. And in spite of this co-culture separated by this permeable membrane, no functional transfer of Cas9 was observed. Now, I wouldn't be telling you this if there wasn't a silver lining to the story. And that came when we did the next obvious experiment, which was to co-culture the cells together with no impediment, allowing cells direct cell-cell contact. And when that's done, the signal that Song and observed is dramatically enhanced. Let me explain the various pieces of data on this slide. The setup that he used is to use as a donor cell a cell called HEC293. This is a cell that we typically use for the production and purification of extracellular vesicles. So this is the cell that has the Cas9 construct. He also then set up reporter cells in various cell lines, U2OS, MB231, et cetera, or HeLa cells. And the breast cancer cell line that I've described before, when it's used as an acceptor in conditions without or with biotin, showed a nearly maximal signal of N loop to F loop. So over the course of 6 days of co-culture, when the cells are allowed cell-cell contact, HEC293 cells can functionally deliver Cas9 to MB231. Now, one piece of data that's clear here is that HeLa cells are not good at receiving this material from HEC293, but that's not the only failed pair. When HEC293 cells are set up as both the donor and the acceptor and co-cultured for 6 days, no signal is observed. They are incapable of transferring among themselves. Conversely, when MB231 cells, which are a robust acceptor, are set up as the donor for transfer to HEC293 or for transfer to itself, this cell is a very ineffective donor. So there is some asymmetry that we don't yet understand, where a certain cell type can be an effective donor and another cell type can be an effective acceptor. What could this mechanism possibly be that would allow an otherwise cytoplasmic protein to be transferred between cells? Well, there is a literature going back some 20 years on the formation of intercellular junctions called tunneling nanotubes. These come in two flavors. One is referred to as an open-ended tunneling nanotube, where a projection from one cell approaches, touches, and then fuses with the target cell to create a continuity for possible traffic one way or both. There also is another major example that's been characterized and found to have some actual physiologic role, and that is a closed-ended contact where a tubule emanating from one cell touches the surface of another cell, possibly forming a junction of some sort, but transferring, for instance, a ligand to the surface of the acceptor cell. These have been called cytonemes. Now, for those cells that form an open-ended structure, it's known that actin filaments project across this tubule, and other studies have shown that complexes, proteins, even organelles can transit from one cell to the next, sometimes very slowly, but sometimes very inefficiently. So we sought evidence to test the possibility that the Cas9 transfer we observed was dependent upon tubules of this sort. First of all, we showed that actin is essential for the transfer. If you do the co-culture in the presence of sublethal concentrations of inhibitors of actin polymerization, we see no such transfer between cells. We also considered the possibility that the acceptor cells were somehow using endocytosis once brought together by these tubules to receive vesicles from the donor cells. But inhibitors of endocytosis and knockdowns of genes such as clathrin and adaptor protein had no significant influence on the traffic of material from one cell to the next. Well, then, of course, we had a close look at cells co-cultured under conditions of efficient transfer. So let me set up this image for you. Donor cells in this case are bright green. They are expressing a high level of Cas9 GFP tethered to CD63, as I've indicated. The acceptor cells are labeled in this case by incubating the co-culture with a fluorescently tagged form of wheat germ agglutinin. Wheat germ agglutinin binds to glycoproteins on the cell surface and is internalized, and it populates endosomes and lysosomes. This is happening on the donor cells as well, but that signal is overwhelmed by GFP in this particular filter to make this picture. So we see in the course of these co-cultures clear junctions between cells, and in a higher mag view you can see this appears to be an open-ended junction. I'll show you an example of a closed-ended junction in a moment. But these form, they come and go. Sometimes they are very labile, they form and then they break. Sometimes they are quite stable. Now let me show you a little video of an example where Song was able to see the moment where this tubule makes contact with an acceptor cell. You see the junction now. You see the appearance of material that seems to be moving back and forth. And then near the end of this video, I think I won't take it the whole way, but this junction breaks and the cells separate. But you can see these coming and going. Sometimes they are very thin tubules, sometimes they are very thick tubules. Now, when Song established a co-culture where both donor and acceptor are HEC293, he observed contacts made between the cells, but they appear to be non-productive. There appears to be a log jam at the point of contact. And let me show you some enhanced images. So here you see a tubule that has formed a clearly very tight junction. And over the course of sometimes several hours, this junction persists and does not resolve to membrane fusion very, very frequently at all. So that's seen in other situations where no effective transfer is observed, and yet a junction is made, likely because one or the other cell is lacking an essential fusogenic protein. Now, we looked at a number of possible genes that could be involved in this productive transfer, and the one most interesting gene that turned up is the gene that encodes a protein called dynamin. Dynamin is a protein that has been demonstrated to play a role in receptor-mediated endocytosis. It forms a ring about the neck and facilitates the pinching of a vesicle from the cell surface into the cell interior. But I told you that we can block endocytosis with other inhibitors and see no effect on the functional transfer of Cas9. And yet when dynamin is knocked down, specifically in the acceptor cell, MB231, there is a nearly complete block in the transfer of Cas9. Interestingly, and in contrast, when the donor cell has dynamin knocked down, there was only a marginal effect on the transfer. Thus, we suggest that dynamin has some role in the acceptor cell, possibly in the traffic of an essential membrane protein from the trans-Golgi to the cell surface of that acceptor. That protein would then be required for productive fusion of the tubule emanating from a donor cell. There is evidence in the literature that dynamin not only serves in endocytosis, but serves a role in the delivery of vesicles pinching from the trans-Golgi apparatus. Let me show you an example then of a junction formed when the acceptor cell has dynamin knocked down. It's very much like what we saw in closed-ended contacts formed between HEC293 cells. So here are close-up images of that junction. Over many minutes at this stable junction, nothing seems to be transferred between cells. Okay, well, let me bring this to a close with a summary of this part of the story. We succeeded in the first instance in creating a vesicle, an exosome, that has an enriched supply of Cas9 and guide RNA. And yet that vesicle is not functionally taken up from the medium or from purified vesicles into acceptor cells. Instead, we feel that this endosome, or Cas9 that is released from its bound contact with CD63, may, through the intervention of actin filaments, progress into the acceptor cell where the Cas9 gains access to the nucleus, edits the target site in front of the N-loop gene, and allows N-loop to be expressed quite abundantly. We favor the notion that the membrane of the acceptor cell must have a special content of some membrane protein, maybe a fusogen, that is delivered by exocytosis from that membrane, but of course we have no idea what that fusion protein or indeed other proteins may be. So this is the very beginning of this emerging story, which we're now quite excited to pursue. The virtue of the approach that Song-Yan has established is that we have a very sensitive reporter for the receipt of an editing function from a donor cell, a reporter that would allow us to use a screen, an siRNA screen, for instance, to look for other genes in addition to dynamin that are required for this functional delivery. Now, our hope is that we can identify the relevant fusogen molecule. There are some candidates that we're testing. And then if we can find such a relevant fusion protein, we may see that it's lacking in the exosomes that we've purified from the donor cells, but we may be able to enrich that missing protein in exosomes and thus produce exosomes that could deliver Cas9 functionally. That, of course, remains to be seen. Finally, again, the picture, the group as of a couple of years ago, the rest of the folks that you see here are busy working on exosomes and protein sorting, other stories that are emerging from the lab. But I want to highlight both Lu Song and Song-Yan, who did the lion's share of the work that I've described to you this afternoon. So with that, I'll stop my screen share and open the floor to questions.
Thank you so much, Professor Schekman. That was simply incredible. And I really liked how you kind of started from basic research, went to applications, and it turned out the applications brought you back to basic research, and you're just going back and forth. And I found that really cool. So we'll open to questions now. So questions from YouTube, please put them in the live chat and I will pose them to Professor Schekman. If you're on Zoom, please either type them into the Zoom chat or use the raise hand function and I can let you ask your question yourself. So we can start off with a question from the Zoom chat. So this is from Callan and it's relating to the first part of the talk. And the question is, considering the canonical view of cell cycle regulation, cyclin D upregulation should result in the stimulation of the cell division program. This seems to be slightly at odds with the apparent role of cyclin D in stimulating differentiation, which normally is associated with the halting of division. As such, I'm wondering what you think the role of cyclin D might be in differentiation and why it's specifically selected for this unusual role.
Well, in fact, we do see in the course of this incubation with vesicles, we do see an increase in cell number as well as differentiation. So it may be that this serves to produce more cells that are then entrained into a pathway of differentiation. I agree with the question though. There would seem to be a conflict between differentiation and cell division. And that is a mystery that will remain unresolved for the time being. So what I've given you is just what we've observed. Now, how to interpret that is another matter.
Yeah, thank you. I'll go for a question of my own, but please do keep putting your question into the chat. So you talked about how these exosomes in the developmental context might be used for sort of communication. And have you seen communication of this sort in a non-developmental context using exosomes?
There are many examples of this in the literature. So let me just briefly tell you about my concerns. There is this field of extracellular vesicles and functional delivery is full of a lot of claims. I think some of which cannot be justified. And the problem in the field, one of the problems that I felt strongly about when I started is that most people who work in this area take a cell culture or take a bodily fluid and then concentrate particles from that solution using techniques that do not distinguish between vesicles or non-membranous particles. And so people take a crude pellet fraction and they inject it into an animal or they expose it to a cell culture and they make claims about what this material does, but they really can't ascribe that with any confidence to vesicles and certainly not to the subtype called exosomes. So we have, when we started on this, we made a point of devising this buoyant density procedure to isolate vesicles and to distinguish them from particles. So I I would be cautious about interpreting other results. Now, we have seen in at least one other instance that buoyant density purified vesicles isolated from skin cells, from keratinocytes, will stimulate the rate of wound healing in a wound healing assay, where you take a plate of cells and you put a scratch across the surface. And if you observe the edge of these cells, they migrate into the zone from which the cells had been removed by abrasion. And we find, and this bears much more work, but we find that extracellular vesicles isolated from the keratinocytes, or from differentiating keratinocytes, enhance the rate of that growth several-fold. So maybe there's some other situation where it's not really differentiation, but it's promoting migration from an otherwise more quiescent state, contact inhibited state. I guess
that's really cool. Yeah,
that may be a form of differentiation. So,
Roberta's got her hand raised. So, Roberta, would you like to ask a question?
Yes, thank you very much for the very inspiring talk. I had a question related to the first part of the talk. So I was wondering, so the cell cycle, the cyclin D1 is a cell cycle regulated gene in terms of gene expression. So there is a time within the cell where it's not present generally. And I was wondering if that is reflected within the vesicles. Do you have evidence that there is a time where you don't find cyclin D1 within the vesicles because it's not expressed? Or is there a separate pool of cyclin D1 that sticks to the vesicles regardless of the general, gene expression program in the cell?
Well, you'll have to come to my lab as a graduate student to study that. We haven't considered that possibility. It would be the cell cultures that we're using are asynchronous. They're not. We didn't phase the cell cycle, so we can't say whether, as you predict, the exosomes would only include cyclin D1 during a certain period of the cell cycle. And in terms of subfractionating vesicles, that's really difficult, because you would, in order to subfractionate the vesicles, you'd have to have a marker on the surface of the vesicle that would distinguish different types based on the time during the cell cycle that they were produced. And I know of no such marker. We use markers that are sort of characteristic of the vesicle, but not selective for the point in the cell cycle where they are made. So that would be tough. That would be the first question is feasible to answer. The second question, not yet.
Thanks.
Thanks, Roberta. Thanks, Professor Randy. We have a question from the YouTube chat from Abhimanyu. The question is, how was dynamin identified as a factor of importance in the acceptor cell? Was there a particular screen used to identify this, or did you just try knocking down genes associated with endocytosis? Yeah,
we did. We made a small collection of genes that were potential targets, and dynamin was stood out as the most interesting one. We included that on the list because it's ordinarily considered to be required for endocytosis. I mean, it is required for endocytosis. And yet the knockdown of dynamin gave a much more dramatic effect than the knockdown of clathrin or of an adapter protein. And we've done other experiments that I didn't describe that suggest that acceptor cells, when incubated in the presence of dynamin, are diminished in their export of a set of glycoproteins to the cell surface. It's probably a complex set. And in the literature, there are examples, for instance, VSVG protein, which is a standard marker that people use to measure traffic in cells, is not exported to the cell surface in the presence of dynamin. So I'm afraid that doesn't narrow the number of possibilities down too much as to which membrane protein is required. So what we're really focused on right now is there's one very strong candidate for an endogenous membrane fusogen. It's called syncytin. It's a product of evolution that we borrowed from retroviruses 50 million years ago. And that's endogenously expressed, but its function thus far in the mammal has only been shown to play a role in trophoblast differentiation in the early embryo. So it's not clear what function that protein would serve, in spite of the fact that it is expressed at a low level throughout the body. It's in the cells that we've looked at. So we're really keen to knock that gene out, and we've just finally succeeded in doing that. So we're now testing whether it plays a role in this. So stay tuned.
Yeah, fingers crossed. Thank you. Another question. So why would this method potentially be better than the current methods of delivering Cas9 and gRNA to cells?
Yeah. So the problem with Cas9 delivery, and there are means of doing this, is it's a one-shot program. Because once you inject that, unprotected, you develop an immune reaction against it. So the idea of using exosomes, and this is being considered by lots of people in biotechnology, is that it's a natural vesicle that is in our body all along. So it should have some immune privilege. So it's a cloak that would allow one to design a way of delivering it specifically somehow. So that's the idea. And since Jennifer Doudna is a colleague of mine, she encouraged me to try this. I confess that it's the first kind of experiment that I've ever done in my career that had a potential practical application. But as you see, it's turned into a basic science program with which I'm more comfortable anyway. So.
The next question I think sort of connects to that point about like doing your research with sort of blue skies, curiosity-driven questions or applications, and how you kind of transition between the both. What your thoughts are?
Well, I'm a basic scientist. I've always been driven by just my wanting to understand how a cell works, and I never, when I started now over 45 years ago working with yeast to uncover the pathway of secretion, I had no idea or frankly any interest in whether that would have any practical application. I just figured that yeast was a good model to study. Paul Nurse had just isolated his CDC mutants in Pombe, and it was an obvious way to study a pathway that likely was conserved, and that's the only interest I had. Now, it turned out quite unexpectedly that once we demonstrated that yeast cells have a pathway that is evolutionarily conserved, I was able to help a local biotech company to engineer the expression of human recombinant insulin in yeast. And now that approach produces 1/3 of the world's supply of human recombinant insulin for diabetics. And that was an eye-opener for me. It didn't convince me to do that practical work in my lab, but it certainly encouraged me to collaborate with biotech and to consult for biotech companies. And so my feeling is that we in the academy, we in universities, should be focused almost exclusively, certainly primarily, on basic science. And that's because everything that we do here should be publishable. But in the private sector, of course, the motivation is entirely different and not necessarily leading to publication. But what we do is we take government support to discover the natural world. And so I have been and continue to be completely immersed in basic science.
Thank you very much. That was a quite balanced view. Maybe a final question. Unless anyone else has burning questions, please do put them in the chat or raise your hand now. But otherwise, a final question. So the tunneling nanotubules, which you've kind of come to at the end, what was their sort of evolutionary function, and what do they do in nature? Do we know?
I should have emphasized that open question. These structures have been seen in cell culture, in a variety of cell cultures. But they have, the open-ended structures have not yet been demonstrated or functionally discovered in an organism. in a metazoan. That's the big challenge. Is this physiologically meaningful, or is it some artifact of cell culture? Now, Tom Kornberg has demonstrated that these structures, which he's called cytonemes, operate in signal transduction in Drosophila. And his experiments are entirely in vivo. But his structures, he doesn't think, progress to open-ended continuities. So that's the big question, which has not yet been answered. So here's my plan. The plan is to conduct this screen to look for more genes and to find genes that are required for this process that could sustain a deletion in an organ-specific manner in a mouse. And then to go in and knock the gene out tissue specifically and look for some effect that can be ascribed to a failure in intercellular communication. That's the plan. It's a long-term plan, but I see no other way of validating this in vivo.
That sounds fantastic. Thank you. So I don't think there are any more questions. And in that case, I'd like to thank everyone for coming. Thank you to the audience. Thank you. Over the last couple of days, we've had 4 fantastic sessions. And I'd like to thank Professor Randy Schekman for coming, speaking with us today. It was inspiring. It was just fantastic. And I think it was a great way to conclude Varsity Sci this year. And we just hope to have more of these for the next few years to come. And Professor Schekman, if you ever come down to visit, we would be glad to host you in Cambridge.
Well, I'd love to visit Cambridge again, but until international travel is allowed, I'm sitting here in my office communicating as best I can.
Thank you very much.
All right, great. Have a good weekend.
You too. Thanks.
Bye.
Bye.